Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa. 안녕하세요, and hello all. Welcome and thank you, SynBYSS people, for joining us the 186th seminar, and I call it a KAIST Day or Korean Day. I am in Korea and today is the 12th day of my 17-day nonstop trip. Last week, I attended, chaired a session, and gave a talk at the inaugural Microbiome Editing Gordon Research Conference organized by David Bicker and also Jennifer Doudna, a Nobel Prize winner in 2020. This week, I attended Korean Space Bio Conference and visited KAIST to give a seminar hosted by today's pioneer speaker, Professor Kim, and also to meet many colleagues, including Professor Sang-Yup Lee, the Vice President of KAIST and SynBYSS pioneer speaker at the third webinar in 2021. Although I have slept 3 to 4 hours of uh, probably almost every day since January 11 when I embarked on the trip. It has been so great and exciting to meet and discuss many topics with many old-time and new colleagues and the Nobel laureate, of course, and the KAIST vice president, and then future leaders, most importantly, during 2 university seminars and 2 conferences. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Hyun Kim. He is a professor at KAIST, and I have known him for many years. He is much younger than me. I'm talking about age, but his publication record is truly remarkable and amazing. I would say his publication record is better than mine, and I was so glad when he accepted my invitation. Hyunuk, thank you so much for your contribution to the scientific society and the invitation and hosting my seminar at KAIST this week and staying with us, although it is already after 1 a.m. in the morning, and the virtual podium is all yours now, and thank you again.
Okay, Professor Moon, first I'd like to thank you so much for inviting me and giving me a chance to connect with the new people and also share my thoughts with people with the community. So, so I believe you can see my slides.
Yes.
Yes. So my title is very simple, synthetic biology and AI. So as you can see the title, I'm a computational guy. So I like to discuss synthetic biology from the perspective of AI. Now, before I begin, first, this term synthetic biology, well, it can be interpreted in several different ways. In my case, I often tend to associate it with biomanufacturing, probably because of my engineering background. So I'll be discuss synthetic biology in the context of biomanufacturing, particularly with focus on optimization of microbial production. First, I'd like to share with you my recent experience with AI and like to then move on to some challenges and opportunities that I keep thinking about for some time. What you see here right now, these are the captured images of ChatGPT. So last semester at KAIST, I was teaching numerical methods, and before the semester started, I asked AI whether it could solve some math problems because I was aware that AI was already good by that time. So what you see on the screen, the left one is the regression problem, and the right one is the multiple equation problem. Well, AI was to solve them quickly and correctly. I was like, damn, what am I supposed to teach? I tested a few other problems in a textbook, and the AI was managed to solve most of them correctly, and again very quickly. So I had to update my syllabus, and I still remember vividly that it was the moment I came to realize that AI is really here with us, and I started thinking we have to change the way we think about the research and the way we do research. And initially, well, literally, just everything that we do in our daily lives. Sorry about the language, the upper image because of it's Korean, but it's having the same message as with the other two images, which I took from New York Times. So we tend to have this sort of unsure anxiety about having this AI with us. The challenge is that so hard to make predictions about. It's hard to make predictions about what's going to happen after about 3 years, because every new week is literally speaking is a new word. We keep hearing about new updates, new models, layoffs, all that sort of things. So I've had some quite of headache for this issue for some time. Then I started figuring out what AI can do and cannot do. This image comes from my recent discussion with my group member. If you look at the image, you see the word SynBYSS. Now, SynBYSS is it's a computational way of presenting molecular structures by using string of characters. Because I was able to solve math problems already quite accurately and quickly, I thought this was no problem. But then, to my surprise, I realized that AI could not generate smiles of this molecule, which I captured in a paper. It was like, wow, moment. Okay, so looks like AI cannot do this properly. And when I tested a few things during the course of data collection for my AI model project, we found out several other things that AI cannot do. And which means there are quite a things that we human can still do, which I thought to be quite promising. So these are a few examples that has happened to me last half a year. Just wanted to share it with you before I move on to AI thing. So that was kind of introduction. Um, for my group, we do systems biology and computational biology. Our objective is to develop computational models by using a variety of biological data, mostly omics data, but also increasingly papers. And because we develop computational models, our objective is very much focused on making predictions rather than analysis. In that regard, what we do is quite different from a traditional bioinformatics because we have to make predictions. Ultimately, my hope is that we contribute to minimizing time and cost of large scale experiments in synthetic biology and in medical studies. We implement largely, we take two main largely we take two different approaches. One is obviously machine learning or AI, and the other one is numerical methods. If you see the if you see this cursor, this network optimization, it corresponds to numerical methods. Today, of course, I will focus my talk on machine learning. Within the lab, we pursue two different topics. One is synthetic biology, which is a topic of my today's talk. The other one is systems medicine. Once again, synthetic biology, we often deal with it in the context of biomanufacturing. Systems medicine, we are very much focused on drug targeting. We've worked on cancers, dementia, and viruses, infectious diseases, but I won't talk about them today. For synthetic biology, our main interest is on microbial natural products or secondary metabolites. So we are very much interested in studying a special group of microorganisms called SynBYSS. We consider them to be an important source of novel medicinal molecules such as antibiotics, anti-cancer molecules, and immunodepressants. Um, they are very potential resource of such valuable medicinal molecules, but nonetheless, there is a highly complex biology behind. That's where computational biology comes in. So this is the main topic within synthetic biology, but of course, we also touch a few other things, and I like to share with you some of them during this short talk. So first, I'd like to talk a little bit about data generation. So in this AI era, everyone talks about AI, but to my uh, knowledge, people do not often get to talk about data itself. AI is more or less data. So we should really also pay attention to the quality of data that we get to deal with for AI model development. If time allows, I'd like to quickly go through some of the AI models that we developed in my group. So we see increasing amount of biological data. This is a bit outdated graph, but still, I think the overall trend is still holds valid nowadays. And we are witnessing all sorts of AI models across different biological layers. Now, this may make us think that now is a truly big data era, but in fact, it is not. We are still living a data hungry era. So whenever we want to develop AI models, we often visit, representative databases. This is the starting point often the time. But the problem is, if they are available for our AI objective, that's good. We're lucky. Often the time, there are no data that we want to address our specific given problem. We have to generate the data in-house. So that's the case. Even if the data are available for AI model project, um, that's just the beginning. The thing is, often the public data we get to use, they have all sorts of noise, errors, dirts. The thing is that if we want to make good predictions, high quality predictions from AI model, the data quality should be good enough. But that's not really the case. Garbage in, garbage out. That's very famous cliche. Um, there are many noises, errors. One typical problem would be data imbalance. So that's really compromising the quality of AI models to be developed. So one important activity in my group is to create benchmark data set. So we are collecting all sorts of data in a cell specific manner, and we are cleansing the dirts, and we're pre-processing them, and we want to create benchmark data. When I say benchmark data set, it is a standard test set that we can use to evaluate the quality of existing AI models and new AI models to be developed. And also we can use them to evaluate any new data to be generated. This is a very simplified scheme of our ongoing activity on creating benchmark data set. We want to automatize many of the steps involved during this data pre-processing process. We are pursuing this activity in collaboration with the Korea Biofoundry. It's a big initiative now here in Korea. Our hope is that once we have this benchmark data set ready, we want to release them through a public repository, and then we want to manage them, update them in a systematic and in an automatic fashion. So I hope, because we are now collaborating with the Biofoundry initiative, our hope is that we deploy such benchmark data sets to evaluate new data that will be generated from the Biofoundry, and also new AI models to be developed. I've so far discussed the AI, the both issues and the potential opportunities. But this message goes to many experimental people as well. Simply, it's because now it's an AI era. It doesn't mean that experimental research is not important at all. Well, rather, I should say experimental research is getting even more important if we are pursuing really high quality research. If we can generate scientifically meaningful outlier points, then I think that's really worth doing. Simply, um, just as long as the research is really good, it's not routine and it's not something that cannot be replaced by AI, then I think this such pursuit of high quality research is it will still be very, very important in this AI era. I think, Professor Moon, I think I spent already 10 minutes. So probably I spend 1 minute or two, then I'll finish my talk. So we've been developing all sorts of AI models. The first one is about the impacts on cultivation profiles on gene expression. So this has something to do with the fermentation process. So we initially developed this what is called deep MGR, wanted to predict the effects of cultivation environment on gene regulation. So this was a kind of warm up research. We are now collaborating with the company. I'm very much interested in doing really impactful work with the company. So we've been developing a more fine-tuned, more sophisticated AI models that can boost the productivity of fermentations that are being done at the companies. We are also developing similar models, other different types of microorganisms. That's the idea. And then we've been very much interested in what is called literature mining. So if you look at the paper, we can see this sort of a highly sophisticated, complex pathway images. So we developed a AI model, what is called ABPI, which basically extracts, information from pathway images from many number of papers. When I say many number, it's not just 10 or 20, but tens of thousands of papers. Um, so the output is this sort of table if we provide this sort of image to the eBPI AI model. Um, we did the quite tough uh, testing for this eBPI AI platform, and that we were able to extract information at accuracies that lie somewhere between 50 to 75%. Um, what's important impact about this ABPI is that we can actually extract information that is not still available in main public databases, but only available in papers. And we can extract such papers from from large number of papers. So that's the idea. AI can, of course, can do it, but I mean, AI cannot handle tens of thousands of papers. So what we want is on-site um, AI model that we can deploy in a biofoundry platform. So that's the idea. So we are currently developing updated version of it, hoping that we can generate cell-specific information by using this updated version of ABPI. So this is a summary, but uh, uh, I only talked about AI, but then mechanistic models, physics-based models, chemistry-based models, they are also equally very important. They are complementary to AI models. So we should also think about such computation models depending on the problem definition and also availability of data sets. So I spent a bit longer time, uh, but then thank you so much for your attention.
Fantastic talk. So now I conclude you are much younger than me, but you also even speak much better than me in English, even though you never lived in English speaking country, if I remember correctly.
I
So,
spent 1 year in California before.
oh, you did. Okay. Okay, but by the way, so I mean, I living in United States more than 20 years, so fantastic. So I 100% agree with you. The quality data is very important. And while I'm listening to your talk, I realized the AlphaGo,
Mm-mm.
the beat human, the especially the human Sedol E, the
Yeah,
Korean Go player, and
yeah.
I actually used to play Go very well, and that is a very
Cool.
shocking moment to me. But I realize now the smile case you show the failure of the AI,
Mm-mm.
because I don't believe the AI trained with the good quality data, but AlphaGo trained with the best, play the play kind of record,
Mm-mm.
thousand and 10,000 of the good, very fantastic, record. But probably Smile, if we train that one with that good quality data, I believe one day also they excel. And then in fact, the Shadow Lee won one game with the
Mm-mm.
very weird movement. Uh,
Mm-mm.
that is a never any player, professional player, never thought about that type of dumb or weird play move.
Mm-mm.
But that is kind of make the AI kind of confused, and then actually AI kind of
Mm-mm.
collapse at that time. So it remind me of a lot of different things, how we go with the AI in the future and how we're gonna
Mm.
improve, etc. So thank you so much, such insightful, talk today.
My pleasure.
All right, so now the main speaker of today with a longer introduction, Dr. Jaeseung Hahn is an associate research scientist in the lab of Cam Leong and Tal Danino, another SynBYSS pioneer speaker at Columbia University. His work focuses on synthetic biology approaches to engineering medicine, from repurposing DNA as a building block for nanomedicine to programming bacteria as cancer therapy. He earned a PhD in medical engineering and medical physics from Harvard MIT program in health science and technology under the supervision of William Shi, and also a BE in biomedical engineering from Macaulay Honors College at the City College of New York. Jaeseung is currently supported by the NIH Pathway to Independence Award, so-called K99R00 from NCI, one of the most competitive and prestigious young research award. He is also Korean, and I should call today a Korean Day. In fact, this is, I believe, the first time when all participants are Korean, although it is already the 186th webinar. I believe it shows Korean synthetic biology has advanced tremendously for the last 5 years, thanks to all the young researchers and also big names. Um, Also as other area like economy, K-pop, literature, and culture have advanced with global recognition. I'm very proud of being Korean, and also very happy to be Korea today. And I'm also honored and very proud of meeting these young future leaders uh, this week during my trip. And Jaesung, uh, I'm meeting you today virtually, but we met in the last year, I believe. Uh, Thank you so much for your time today, and please take it away. Thank you again.
Thank you so much for a kind introduction, Professor Moon. And and I will I'll continue with my introduction. Hi, my name is Jaeseung Hahn, and I'm what you can call a classically trained biomedical engineer. So I did my bachelor's in biomedical engineering and PhD in medical engineering and medical physics. And also I'm doing my current postdoc in the Department of Biomedical Engineering. So what that really means, because biomedical engineering is an interdisciplinary field, and I studied breadth of physical sciences and engineering fields like physics, chemistry, biology, chemical engineering, electrical engineering, mechanical engineering. So all for the sake of understanding and engineering for human health. So my main subject matter is human, like myself, and you can approximate humans to be about 1 meter in size. But the basic unit of life, cells in our body, are on the order of 10 micron. And of course, we're not alone. We have many microbes living with us, and they're on the order of about a micron. However, we can look closer, and the basic subunits of life are really the biomolecules like nucleic acids, proteins, at the size regime of nanometer. So we can, of course, go further, but we get into the realm of chemistry and physics. So we need to be able to observe and manipulate biology at the nanometer scale to operate and intervene at its finest resolution. So as an undergrad, I naturally got into research in nanomedicine, worked on synthesizing what you see, the gold nanostars, what we call. It has interesting optical properties that are useful for diagnostic and therapeutic applications. While I was able to make these beautiful structures out of gold and its fine features reaching below 10 nanometer at this branch tip, but I was not quite satisfied with the fact that the nanoparticles are all different in size and shape. And if you look at biology, they are really, really good at making this tiny, tiny structure that exquisite details and with a function. So as I was thinking about what should I do during my graduate training after finishing my undergrad, I learned about this field of DNA nanotechnology and a technique called DNA origami. So it folds a long single-stranded DNA, scaffold strand, usually from M13 viral genome. And with short synthetic DNA staple strands shown here in red, they're complementary to different parts of this scaffold strand. You can essentially fold this DNA, long DNA structure into specific structure that you designed. So much like the art of origami that folds paper into different shapes. So using this technique, Paul Rothman in 2006 showed that one can fold DNA into arbitrary shapes based on the design with atomic precision. For example, it is possible to fold DNA into what he called a disk with 3 holes that resembles smiley face, which is about 100 nanometer in diameter, and its narrow part of its mouth being the thickness of 2 DNA double helices, about 5 nanometer. So for my PhD, I joined the lab of Dr. William Shi, specializing in constructing DNA, 3-dimensional DNA origami structures. And at the time, we were developing this DNA barrel structure shown on the screen that we could use as a molecular breadboard for integrating different biomolecules. And this barrel structure is about 30 nanometer in radius and 24 nanometer in height. And this red dots that are shown are this addressable pixels that we can put whatever molecules that we want to. And we can also look inside by making a cross section. Then we also have shown in green different pixels that we can address with different gas molecules. So it has an inside and outside. And you can also see that in using electron microscopy image, we were actually able to fold this structure as we designed. And this is we thought that this would be very useful in terms of shape as well, because we were able to form homodimer by just stacking these 2 barrels together, and even tetramer. And essentially, this is a modular system. We can build more and more complex system. And when we were trying to think of, okay, we have this molecular breadboard, what should we do with this? We kind of looked back into nature for inspiration, and we saw vaccinovirus, which, unlike most other viruses, they have their own RNA polymerase, which allows them to actually replicate in the cytoplasm instead of having to enter nucleus and take over the host transcription machinery. And we wanted to kind of recapitulate this mechanism and build what we call RNA producing nanofactory. And shown in this picture is a cross-section of our barrel structure. And then we imagine that we can pack this long DNA barrel structure with different kind of functional units. And what they are is essentially the DNA leash, circular DNA leash that will catenate the circular DNA template so that it can be transcribed by RNA polymerase to make this concatenated RNA transcript. And this multimer of RNA transcript can be processed by RNA processing enzyme into monomer for downstream function. So in order to do this, we of course start with our barrel structure. And just for this talk, I'll cut this barrel structure, flatten it, and resize so that it's more manageable to kind of look at what it looks like in terms of design. And I was telling you about these addressable pixels earlier, and they're essentially this single-stranded DNA handles we call with a very specific DNA sequence, so that whatever gas molecules that we want to introduce, they just have to have anti-handle that is complementary to this DNA handle. And of course, we had used a RNA polymerase specifically from T7 virus, the T7 bacteriophage. When what we did was that we fused it with a snap tag so that we can introduce the DNA strand that is complementary to the DNA handles sequences. And for the processing enzyme, we use the CAC3 ribonuclease that can recognize specific sequence and then cleave exactly, so that we can, once we produce the concatemer of RNA transcript, we can cut them into monomers. And of course, we also had this catenated DNA template in the leash. We had the RNA polymerase binding site, so the RNA polymerase can be bound to the template and then do the transcription. DNA pane is for I'm not going to talk about today, but for the quantification of the parts, and also the origami binding sequence so that it can the template can actually be fixed onto the DNA barrel structure. And the DNA template itself had a T7 polymer so that it can be recognized by RNA polymerase, CSC3 substrate, so that the RNA can be processed once they are transcribed. And then we put the target RNA. In our case, we used a Berkeley aptamer because we can observe them using fluorescence. So we went on to put all these things together. We make this design the tetramer of barrel structure, and this structure had 12 RNA complex catenane DNA templates. So this is our RNA processing unit. And then we also programmed 96 CSC3, so RNA processing unit, into the barrel structure. And to test this, in experimental, we actually built 3 kind of variation of this. So the first one to the left is the DNA barrel structure that only have the RNA production unit. So when I say handle minus, that means that the structure does not have handle for the RNA processing unit, the CSC3 protein. So without that, there is no RNA processing unit in this nanofactory. The second structure that we built was RNA. It has all the handles, so it contains both RNA producing and then processing units. But we introduced, after assembling the whole structure and then and getting rid of all the freely floating molecules to get the pure our DNA nanofactory. And then we introduce a strand displacement strand so that it can free the bound CSC3 protein to test out whether or not it's actually necessary for this processing and production units to be co-localized. And of course, the last structure all the way to the right is everything packed into our DNA origami structure. And to look at if the structure, this nanofactory, are actually producing and processing RNA, we can look at it using electrophoresis. So what it is that we can load the sample at the top of the gel, and we apply electric current. And because nucleic acids are negatively charged, they are pushed down over time. So if they're monomeric RNA, so meaning that they're produced and then also processed completely, because they're lighter, they get pushed much further compared to, for example, if in our case of nanofactory without any processing unit, we would produce this a catamer of RNA that is much heavier, therefore will travel much slower. And what we expect for the other two structures to the right is that because they have this RNA processing enzyme in the solution, should be able to process and cut this large RNA into smaller subunits. But because the middle structure that we free the CSC3 protein, they are not able to colocalize, and therefore its processing efficiency will be lower than the structure all the way to the right, where everything is packed into one structure. And so we actually ran this experiment, and we calculated the RNA processing efficiency by looking at how much of the RNA is actually being processed from the total amount of RNA in the gel. And we can look at, we saw that for the structure that did not have any CSC3 protein, as we would expect, we didn't see any processing of RNA. But for the other two structure, we did see processing, but significantly, what we saw was that when all the components are actually co-localized, it had about 20% higher processing efficiency. And we also looked at this in electron microscope, and this was quite amazing when I first saw it, especially the very first to the left. I saw RNA being spooled out, kind of like a mushroom, out of our nanofactory, and that was really amazing. But what we see all the way to the right is that we don't see this mushroomy structure anymore, because they're being actively processed as they're being produced. In the meanwhile, in the middle, when the RNA processing enzymes are not co-localized, we still saw this RNA structure kind of coming out. So when we finished this work and submitted for publication, and one of the reviewer actually commented that this work really pushed the technology to the limit. And of course, it's quite amazing to hear this type of comment, flattering, but also at the same time, as a researcher, you kind of start wondering, oh, like if this is pushed to the limit, like what next? And I was thinking, of course, there are a lot of different ways to push the limit. I think we can continue to work on the same topic and then trying to push through. But I was thinking about what else can I do to really build more complex biological system. And I think this is more kind of closer to what the audience do oftentimes, looking at the cells. We already have cellular systems that are vastly complex, and a lot of times what we do is that we can change the DNA sequences to reprogram them and do some really cool stuff. And at the time, when I was thinking about moving to more complex cell, mammalian cells seem a little scary, but bacteria seem simpler, a bit naive assumption, in that bacteria, of course, still very, very complex. And but other kind of cool thing about bacteria is that they have been used as cancer therapy for quite a while, at least in modern medicine, as long as over a century. And even though they kind of fell out of the favor due to invention of radiotherapy and chemotherapy, also 100 years ago, there was no antibiotics. So there's a certain risk. But now, hundreds of years later, we have a lot of different knowledge and also technology available for us. And then a lot of people are actually thinking about how do we use bacteria as a therapeutic, and specifically for cancer therapy. There has been quite a bit of interest recently. And the reason is that you can see this bacteria as a kind of perfect machine, micro-sized machine that's made out of nanometer scale components. And they can really do a lot of things. They can sense molecules, they can move, swim through solution using flagella. And of course, they can also produce things by sensing the environment, either reporter proteins for diagnostics or even therapeutic proteins for actual therapeutic applications. And the kind of vision in the field is that we can use this bacteria themselves as a therapeutic to colonize a tumor and produce a therapeutic molecule, and they can work with other existing therapies like chemotherapy or even immunotherapy to really trying to tackle the challenge in cancer. And another interesting is that there are many different niche for bacteria to live. As I was kind of alluding to, they do colonize, some strains of bacteria colonize tumor, but there are a lot of them in gut and skin as well. So there are many routes of administration available. So we can really start to think about how can we really leverage this organism for medical applications. But of course, whenever we think about developing any drugs, we have to think about how do we deliver it. I think that's something that I always kind of stumble upon whenever I'm trying to develop new therapeutics. So say we have this really good therapeutic bacteria developed in the lab. And in order for us to actually use it, we need to administer it maybe through pill form, like how we take probiotics, or we could even imagine that we can inject either intratumorally or even systemically. And in our lab, we actually do a lot of systemic injection in mice, and we are hoping that they will get to the target lesion, in our case, tumor microenvironment. But of course, that's not the only place the bacteria are going to go to. Just like any other drugs, they will have some toxicity related to it, especially because bacteria have a lot of immune reactive molecules, pathogen-associated molecule patterns, and also they are going to be cleared. So you also, at the same time, are fighting to get your drug into the target lesion before they're being cleared away, because our immune system is really good at recognizing microbes and getting rid of them in the healthy tissues. Another kind of interesting thing about this using living bacteria or any living cells as a therapeutic is that they are able to replicate. So once they replicate, especially after they replicate, because this bacteria can get in, they can also get out. So they will leak into the system, and then that, of course, now you have to worry about toxicity, but of course, they will be also cleared away. So in order for us to really try to deliver this new class of therapeutic better, what we would like is that initially, when we administer, it will get to the target lesion efficiently. But once they're in there, we want them to be cleared easily so that it's effective, but also safe when we use it as a therapeutic. And the way we thought about in the lab, how do we do this, is kind of inspired also by what I have been working as someone in the nanomedicine. Whenever we try to deliver nanotherapeutics and we try to change this interaction with the host immune system, we coat the nanotherapeutics, for example, with PAG coating, so that it will be more inert and then not be recognized by immune system. And that's what we envision doing with the therapeutic bacteria. But because it's a living system, we also can control its dynamics. So what we envision is that we can initially protect the bacteria by having this coating. When once we administer it, it will be get to the target lesion more easily. But once they are there and then replicate, they will lose this coating. And then once they leak out, they're recognized by immune system and get cleared more easily. So we worked with E. coli in ISO 1917, which is also commercially available probiotic as under the name of Metaflor for many decades now. And interesting thing about this bacteria is that they possess this thing called capsule polysaccharide. So this is actually many pathogens have this capsule polysaccharide, which protects bacteria from the environmental threat, which includes immune system actually. So this bacteria already possess this kind of protective layer. So we thought that if we can control this protective layer, then we can potentially develop this this dynamic coding system we are envisioning. So we built this a pretty simple genetic circuit. So from the genome, we just knocked out one of the genes that are involved in the biosynthetic pathway of the capsular polysaccharide, and we reintroduced it, but under the control of that guy. Therefore, we can control the expression by introducing IPTG. So without IPTG, there is no capsular polysaccharide, meaning that it will be cleared more easily inside the body. But if we put IPTG to induce this circuit, then it produces the K5C gene, therefore the capsular polysaccharide, and then it helps with the immune evasion of the bacteria. So we built this system, and then we tried to look at this nanocoding, biological nanocoding under the electron microscope. And then to the left is without any IPTG, and then to the right is with IPTG. And then you can visually see that there is this thicker layer that forms when we actually introduce IPTG. And we can quantify this. And without any IPTG, we saw that about, in average, this layer, polysaccharide layer, was 44 nanometer, but with 10 micromolar IPTG, it increased to about 80 nanometer, double the size. And we think that this thinner layer is probably other saccharide molecule like LPS that is also being stained. But once we induce this K5C gene, we see this thicker actual capsule polysaccharide being produced. And of course, we need to look at, does it actually do the job? Does it affect the clearance and immune evasion? And we test this by first using blood bactericidal assay, because blood has these complements that can kill gram-negative bacteria, which is the case for E. coli. So without any capsule polysaccharide, this bacteria will be very prone to dying. But with the capsule polysaccharide, they are protected. So we induce the production of capsule polysaccharide using different concentration of IPTG. And here on the horizontal is the different concentration increasing to the right, and then vertically is more, as you go down, is more diluted. So at the top you can see that it's red because it's blood incubated with the bacteria, and then we dilute it further and further, and then we can kind of see this colony forming units, which we can quantify to the left here in the graph. And then as you can see, after incubating with the blood, without any IPTG, the bacteria are very prone to dying and not form any colonies. But as you protect the bacteria more by inducing with the IPTG, they form more colonies in the plate. And we can look at the dynamics of it. And here in the black is the wild type E. coli Nissl, and then in the light gray is the K5C knockout. So the wild type bacteria actually dies a little bit, and then it can actually sustain its population over 6 hours. But without any K5C gene, they actually die very fast because they're not producing any capsular polysaccharide. And with this inducible system, we can control this dynamic by increasing the concentration of IPTG, therefore the different induction of this expression of the capsular polysaccharide. And also we looked at how it interacts with the immune cells. So if we co-incubate with macrophages to see how they do with the phagocytosis, we see that so without IPTG, without any capsular polysaccharide, they're phagocytosed more. So it's shown in what we do is that we co-incubate, and then we isolate the macrophages, and then we lyse the cell, and then look at how many bacteria are actually inside the macrophages. And therefore, the more colony forming unit we see, that means that there has been more phagocytosis. But with IPTG, because it's better protected, they are also decrease the phagocytosis. And we also looked at how this capsular polysaccharide affect the immunogenicity. And we saw that we do this by co-incubating again with bacteria with the immune cells, and then look at the cytokine production. And we looked at the TNF alpha, which is the the cytokine that's produced, signifying the immune reaction. And with the E. coli initial wild type and with this inducible system, what we call ICAP, when they are induced to produce the capsule polysaccharide, they produce much less TNF alpha. The immune cell produce much less TNF alpha compared to the E. coli initial knockout that does not have the capsule polysaccharide. So what kind of going back to what we have envisioned originally. So when we initially induced this bacteria to have this capsule polysaccharide protective coating, and then inject it into mice, and then once it targets the tumor and colonize, they should lose this capsule polysaccharide and then become more clear more easily in the body. And the way we saw was that without any capsule polysaccharide, there will be short-term high immunogenicity and then short-term toxicity. But with bacteria with the wild type bacteria that always have this capsule polysaccharide, initially they might not have immediate immune toxicity through recognition from the immune system. But in the long term, because they can survive better and then also can spread to other parts of the body other than tumor, they should have perhaps more of a long-term toxicity. So what we think is that using this inducible capsule polysaccharide system, we can both decrease the immediate toxicity as well as the long-term toxicity. And we tested this by injecting first mice with different dose of different bacteria strains that we had. So we had the 3 strains here: E. coli initial, wild type, that always have this capsule polysaccharide on, the knockout, which does not have any capsule polysaccharide, and then the inducible system that initially has its protective coating, but over time it loses. And we did a single IV injection into mouse tail vein, and then we looked at different dose how that affects the well-being of the mice, and we measured this toxicity by looking at how much body weight mice lose from being sick. And in one dose, what we saw was that so with E. coli initial wild type, they will lose about like a little over 10% of the body weight by day 3. And with the knockout, it will be a little less, and then it recovers faster. But with using inducible capsule polysaccharide system, we were able to actually decrease the amount of weight loss as well as kind of recovery speed as well. And we did this experiment over many different doses, and we were able to produce this toxicity curve. And those toxicity curve, and we saw that by using this inducible system, we were able to increase the maximum tolerable dose by 1 order of magnitude. So next we were thinking about, okay, if this system is safer, can we actually make it even more efficacious? So in classical pharmacology, we look at this dose response curve where for increasing dose, there will be increased efficacy as well as the toxicity. And the maximum efficacy we can really achieve is based on, of course, the efficacy curve, but also the toxicity curve. Whatever dose that we introduce has to be tolerable to the patient. The idea is that by using this inducible system, do we actually push the toxicity curve to the right so that we can achieve even better efficacy? So this was our hypothesis, and it was kind of testing this idea in the field that perhaps the amount of bacteria that ends up in the wherever lesion that we want to target, in this case tumor microenvironment, wouldn't matter, because whether we have a little amount to the left or a large amount to the right of seeding density, they're all going to replicate and they're going to reach the same kind of capacity. So they should have similar or even same kind of a therapeutic benefit. And that's kind of like something that I think people were thinking about as a benefit of using this kind of living therapeutics. And we wanted to test, is that true, or is there actual benefit for being able to deliver more bacteria? And we tested this by injecting our there's 3 different bacteria strains into mice that carries tumors. And we also introduced the data toxin into our bacteria so that it has this data toxin can directly kill the cancer cells. So if we're equipping the bacteria with some therapeutic effect. And we injected at their maximum tolerable dose. As I said, ICAP inducible system had like 1 order of magnitude higher. And what we saw in the subcutaneous colorectal CT26 model is that indeed, when we use higher dose using ICAP system, we were able to actually reach much better tumor growth control all the way at the bottom. And we also actually included in the same blue, but in a dashed line, the ECN ICAP low, which was the same strain, but dosed at same amount as the wild type, so 5 times 10 to 6 CFU per mice. And it showed that it is actually the dose dependent effect, not just a strain dependent effect, because these are different strains. And we also test this in more complex genetic breast PIMT MMTV model, and we saw very similar effect. And you could ask, like, why does this happen? And we have some other data showing that so this bacteria that we injected, they had the luminescent signal so that we could actually track them in vivo. And what we saw was that our inducible system that was injected at the MTD, they had much higher luminescence in vivo compared to the other strains, as well as the same strain, but dosed at 10 times lower amount. And you could kind of argue that maybe this is saying that maybe if you put more bacteria initially, then maybe there are more bacteria inside the tumor. But we see at the end of the experiment, we sack the mice and we look at the tumor biodistribution of this bacteria, and then they all have similar amount of bacteria in the end. So I think that there is some level of fitness, different kind of production capacity for bacteria, and as well as there must be some effect in the immune system response based on the different dose that we inject. But it's still ongoing, and we still don't completely understand. But regardless, very interesting phenomena. So today I told you about my quest towards applying a bottom-up approach to engineer biology, starting from biomolecules and to build increasingly complex systems at the interface of nanomedicine and synthetic biology. And in order to imbue the function in the biological system we build, what I have done is I encoded information into biomolecules, in the most case DNA, but also in the protein as well. And I told you about the kind of 2 examples from my work over the years. One, RNA producing nanofactory to the left, and then of course the bacterial cancer therapy to the right. And I think that there is still quite a lot of room for me to explore in the bacterial therapy space at the interface of nanomedicine and synthetic biology. And just a little over a year ago, I wrote this review paper about the exact topic. And what I would describe what I told you about today regarding bacterial cancer therapy as a nanomedicine inspired strategist. So trying to coat the bacteria with this nanocoating and trying to change this dynamic of immune evasion to develop better delivery system. But of course, there are other ways that we can kind of interface bacterial cancer therapy with nanomedicine. We can have a bacteria nanoparticle biohybrids shown as the lower left, and then also bacteria-derived nanoparticles shown to the right. And I think this space is like what I'm really excited about. Also because of my PhD background of trying to design and construct this complex biomolecular system. And I think there is a lot of room for me to explore in this area. And that's kind of where I want to go as I continue my career. And of course, I have to acknowledge all the people involved in the work. Of course, the first work was done in the Shi lab under the supervision of Dr. William Shi, and then the second work under supervision of Tao Danino, as well as the Kem Leong. And of course, all the work that was done with other people, including our collaborator in the ARPAIA lab. And I do also want to just acknowledge that the nanofactory work was done in close collaboration with Dr. Liu Chu, who is now assistant professor at the University of Toronto. And the bacterial cancer therapy work using capsule polysaccharide was done with Dr. Tetsuhiro Harimoto, who was a grad student then, and he will be starting as an assistant professor at Cornell this coming September. So yeah, this has been real pleasure to work with all these people. And yeah, of course, I need to acknowledge my funding source in my grad school, NSF, and then during my postdoc, NIH. Thank you.
Fantastic. Actually, Dr. Harimoto also gave a talk for this series a couple of years ago, I believe. So this is amazing. I have some question, but I want to ask Professor Kim has some question or comment.
So thank you for a great talk. I enjoyed it. One question that came to my mind was, so it's about regulation in the US or any other countries, because now you are dealing with the human body. Now you wanna deploying genetically engineered bacteria. I'm sure there must be some formal procedure evaluation, but what's the status now? Like, is it well accepted in the society, or the in general?
So I'll say a couple things about that. So the bacterial cancer therapy itself, there actually was clinical trial now in like over 2 decades ago in 2000, trying to deliver salmonella. And they actually showed that it is safe to intravenously inject this bacteria into humans. Though, and ultimately it failed not because of the toxicity, but because it didn't show any efficacy. And the strain of salmonella they used, BMP 20009, was like heavily attenuated. And that was probably the reason why, because a lot of the therapeutic effect of bacteria comes from immunogenicity. Is considered kind of like one of the first immunotherapy. I was telling you about over 100 years ago, Dr. William Cawley injected the Escherichia bacteria into inoperable sarcoma patients, and many patients actually saw benefit. But then now like we have over attenuated, and then what we're trying to do is that can we and that's kind of like what my work was about, right? Trying to kind of keep or improve the efficacy, but also keep the safety level high. So that's kind of like where we're at. And in terms of like social acceptance, I will say that whenever I go to especially conferences where there are a lot of doctors, they will tell me, "Are you crazy? Have you heard of sepsis?" And yeah, and I tell them, yeah, that's exactly why I worked on this project as I enter the field, because I want to see if this is actually viable therapeutic option. And I do think that it will be. And also the fact that like our knowledge of microbiome is immense. Like we know that we are not alone. Like we live with the microbes. It's just a fact. And can we actually engineer this microbial therapeutic in a way that is safer and then efficacious? I think that's kind of like a challenge we have right now. And I think that when it comes to like a societal acceptance, I think that especially for patients, right, who are desperate, I think they're just looking for something that will work. And then I think that's what we are really trying to do, right? We don't know what will really work, but we are trying to find whatever creative way that can have any kind of positive impact in their lives and in our society. And I would say that over time, I think that as we show more promise and make sure that we do this correctly and then in a safe manner, I think that that will come with that. That's my thought. Yeah.
I can give a couple more comments. That's exactly what I want to say. For the gut application, also very recently, one company actually doing the probiotic, the initial strain, the clinical trial. I don't know why they stopped. They about to start the phase 3 clinical trial, but unfortunately they didn't continue because of multiple reasons, financial reasons, some other reason, but I don't know what is exact reason. And then so that means FDA at least kind of more generous than any other country regarding using probiotic or NISL or some other thing for the therapeutic applications. Yes, the injecting into the blood or other, that's a little bit more problem because of all the doctors kind of say we are crazy. Yes, we are crazy, but that is the one thing I want to mention.
What about specificity, target cell specificity, and also stability? So how can to what extent can the cells be specific? Can you make cells to target this very small chunk of tumor cells? Uh, that's what I mean by specificity, and also stability. So you are inserting sort of a gene circuit into the cell, right? So then as the cells reproduce, will they be still in the cells when they lose any of it over the time?
Yes, certainly. I mean, that's definitely like people have been thinking about and working on for many years now. So in terms of specificity, um, in general, like the strains of bacteria we use, the way they target tumor is not through actual active targeting, but what I call like replication based targeting. So what happens is that in healthy tissues, as long as the immune system is intact, they're cleared really, really effectively. Macrophages and also for gram-negative bacteria, they're lysed pretty easily by complement, for example. And tumor microenvironment is like such a specific niche where it's an immunocompromised area, which kind of like the reason why they're successful as a disease. But also it creates this niche for bacteria to be able to colonize. So that's kind of like or at least the kind of bacteria that I use. That's the mechanism of targeting. And certainly there are bacteria that can actually specifically target kind of like different molecule signature. As I was alluding to before, like they have locomotive system, they have flagella, and then they can actually swim, and that they have biosensors. So you can certainly do that as well. And then there has been work regarding that as well. But on another side is engineering bacteria so that they become dependent on a specific molecular signal like biomarkers, so that they cannot survive unless the environment is such that, for example, I think from Tal's lab, we have worked on things like containment through biosensing of tumor microenvironment, the low pH and hypoxic area, the hypoxic condition. So that type of work is definitely done. And further, like if you're talking about like specific cancer cells, like we say, like we get into the tumor and then we want to target specific cell, right? I think that's certainly interesting idea, and then I think it should be possible, right? Because you can engineer the interaction. So like the E. coli that I use is mostly extracellular bacteria, so they don't invade cells, the mammalian host cells, but Salmonella and then some E. coli do as well. So you can potentially even engineer that interaction to specifically target certain receptor perhaps that is unique to specific cell types within the tumor microenvironment, for example. In terms of the stability, yeah, that's again another good question and a challenge, right? So a lot of times when we do this type of engineering, we use plasmid, and people have come up with like a stabilizing mechanism to like a toxin antitoxin to increase that, but they eventually lose. And then some other people tried, genomic integration, for example. But I mean, that's not foolproof either, right? There is still kind of challenge of they can still mutate. So yes, that's a concern for sure. And then yeah, it's interesting thought too, because a lot of times whenever we use genetically engineered uh, organisms, people worry about, oh, like what if they kind of gain something crazy and then it goes haywire and becomes dangerous. But like as a scientist, like we always just worry about like, oh, our poor bugs, like will they survive and will they keep this functionality? And that's true. And that I think that there has been a lot of work towards that. And then kind of entangling the bacterial fitness and survival to the production of whatever, or the stability of the gen circuits or whatever functionality. Um, but I think that is something that people have to think about, and then also measure as we develop this as a therapeutic, so that it's a kind of property that we measure and then we can control, and we know what the kind of risk benefit is.
And besides technologies, I mean, it was a bit fun to see your reactions about medical doctors, saying, "Are you crazy?" I had a similar experience. So I was giving this talk. I had to give a talk for the grant proposal. It was about AI-based design of gene circuits, and we said we deploy to we apply for some God-related applications. And one of the referees in the room, he was like, "Are you serious about this?"
You should take that as a compliment, right? As Dr. Professor Moon said, like, yeah, we're crazy.
Yeah, I believe already I told you, I mean, one day, I mean, but before I saying that was that kind of episode I had, the first thing is the Tal Danino's lab is the world-class lab, using
Mhm,
sensor to make the bacteria specific to the cancer.
mhm.
And in fact, I was scooped by his group, so I kind of
Ah,
gave up, and then I'm not going to do
Sorry.
any cancer thing because I'm thinking about like using, oxygen sensor. So we developed oxygen sensor before, and pH sensor, and also even lactate sensor, I believe. And then one paper actually described all of them, and then I think that Nature biomedical engineering, something like that. So
Yeah, yeah, and
that
Tetsuhiro worked on that as well.
yes, yes. So and then
Yeah,
after that, okay, so I developed sensor, but my sensor is not useful for my cancer application. So I dropped that project many years ago. But the second point, that's absolutely interesting because I probably mentioned to you one day I got I mean, not one day, but more than 10 years ago when I started my probiotic project, I got question from general public
mm.
asking, how can you ensure your bacteria GMO not becoming dinosaur? I was speechless, literally speechless, because I have no idea what to say. And then I know the answer now. The answer is if I make my bacteria become dinosaur, I'm gonna become billionaire tomorrow for sure. And then I'm gonna get also Nobel Prize tomorrow because but that's the public, perception because the people watch too many crazy movies, of course, or very and they are very creative in a sense. So I mean that also drive scientists to do something, because I never thought about the AI becoming so powerful right now because when I was young, but although the crazy scientific movies or comic book now becoming a reality, so we need to be very careful in a sense because that is just happening. That is I couldn't couldn't imagine in the past. I'm talking about even 30 years ago or 40 years ago. So this is a very exciting time. Also, we also we need to very careful in a sense.
Um, what about companies? Do you see many companies working on this area in the US? Yeah.
Yeah, definitely. I mean, companies come and go, but it's still active
Mm.
area of research. And actually, my colleagues in Danilo Lab recently published a review paper looking
Mm.
into that,
Mm.
like what kind of like a landscape of papers, patents, and companies, and then kind of like what areas are they working on?
Mm.
Like kind of, I think one thing was like initially as in like past like 3 decades or so, kind of like effort has been a lot more on kind of vaccine development, for example. But then I think it's kind of moving more towards other area, kind of what you're working on, but like in a different setting, like metabolic engineering, immune
Mm.
therapy. So yeah, it's a definitely active endeavor even in
Yeah.
the private sectors. But I
Mm.
think it's something that we like in the field, like we talk a bit about, and I think like with Professor Moon, we talked about it a little bit like last year when we saw each other at the conference as well. Is that kind of like I think even though they're FDA approved microbiotic products now, like that is not just probiotic, right? Like a fecal transplant, and then like they're actually FDA approved pill form to like oral delivery as well. But they're all kind of like a natural consortia type of products, and I don't think still there is no generically engineered microbial therapeutic in the market. I think at least last time I checked. So I think that's like something that we're really as a field trying to actively pursue. Like, can we make it there? And then I think that will open up a lot more opportunities. I think for in the private sector, and of course in the research,
Mm.
academic research as well. I'm sure.
Yeah.
Sure. We have the audience guest. This is the first time I see the somebody not asking question through the online, but asking by speaking. So, Dr. Lee, congratulations your new job. Just speak and then go ahead.
Yes, very, very fun talk. It's very nice to
Thank
finally
you.
meet you, Dr. Han. Uh, I had a, I had a question about what exactly makes the mouse lose its weight. Because I would imagine this cap liposaccharide or polysaccharide uh, could also become a little bit immunogenic, and if that's the case, maybe it can contribute to them losing weight. So I was wondering why this inducible system is actually making it safer for the mouse.
Yeah. So I think for gram-negative bacteria, probably the major immunogenic factor is LPS, lipopolysaccharide, and that kind of like shocks the immune system, and it releases a bunch of like TNF alpha, and then essentially that makes organism like the mice very sick, and then that's the reason why they lose a lot of it. And then if you actually see them in the lab, you can see them visibly sick. Um, so why this polysaccharide like capsule polysaccharide? So I'll say that yes, it's probably possible that our immune system can develop um, like an antibody, for example, against in this case. So the E. coli nasal has K5 uh, capsule polysaccharide, which is composed of a heparin. Um, but the difference is that is it innate versus adaptive, right? So because
Mm.
like LPS is so ubiquitous and it's something that has existed for so long, like our immune system is innately can recognize LPS, and then just kind of like immune system gets activated. Meanwhile, things like K5 capsule polysaccharide that is existing in E. coli nasal is not one of those kind of like a generic molecule that signals, oh, this is bacteria or pathogen. So that's why we don't see it. But you're right. I think that it's not something we have tried, but I would imagine like if we inject this bacteria multiple times, maybe it is possible to develop like antibody against it, and then it takes some time for adaptive immunity to kick in, but eventually perhaps like that will also cause toxicity. And I think that's also kind of like interesting area as well. So the idea would be kind of nice thing about this capsule polysaccharide system is that bacteria have developed like many different kind of chemical composition of their capsule polysaccharide. So it's just K5 capsule polysaccharide is only one of them. Even in E. coli, there are like tens of them, I think. So there is like this chemical diversity that we could potentially try to look into in the future to make sure that, okay, like if it does become target for immune response, and then that will eventually cause toxicity, is there a way that we can change that kind of this protective coating so that doesn't happen? And it kind of goes hand in hand with kind of like what we see with other microbial cancer therapy using like viruses, oncolytic viruses, right? Sometimes they kind of think that it might be like a one-shot thing, or even like some viruses that we develop, like people use in the research, they say, well, this works in the setting, but in human, if it's like if humans have like in population, we have a lot of people who are immune to these viruses, and it's not going to work. And then I think that you could imagine the same thing could happen to like this bacteria. It's like in the future where bacterial cancer therapy becomes mainstream, and then like the population level, we have immunity against like certain things. But yeah, I think that we still have like a lot of room for kind of like changing that by just changing the chemical composition of this capsule polysaccharide. And we can even, like now I'm talking about like natural things, but like we can even imagine like in the spirit of synthetic biology, can we start engineering the composition of this this capsule and then make it more safer and safer? And my question would be like, can we reach kind of the level of PAG, for example? I mean, of course, PAG also, I think they show that our immune system can eventually develop like antibody against it. But that's kind of like a exciting part too, I think.
I see. My question is more on, so if you compare this to the wild type, the E. coli nasal strain, where it always have the cap.
Oh, yes.
And then the E. coli, inducible E. coli will lose the cap and expose the lipopolysaccharide. And at that stage, it will trigger the immune response, which I think will make the mouse lose more weight. But in your data, it seems that the inducible one is actually more helpful to the mouse. So I was wondering why that timing, like,
Oh, yeah. It's all about timing, and I didn't have time to kind of go over the whole details, but essentially, it all comes down to like what time frame are we working on. So when we inject bacteria intravenously to mice, they get cleared on the matter of like a couple hours. And that's because our bodies, even if they're like protected, our body is really good at recognizing these things and just getting rid of them. So a lot of them actually do end up in like, for example, liver, because they have Kupffer cells, a professional macrophage that will eat up any pathogens, and then in bacteria is just like right size, right chemical composition. Meanwhile, at least in vitro, what we have seen is that the dynamic of capsular polysaccharide deactivating our bacteria shedding, it takes about like 4 hours or so to completely shed, and that's in the laboratory setting where they're like actively replicating. So you could imagine that if the gene is turned off, they're going to stop producing. So as they divide, this coating can be diluted very rapidly, but in vivo probably it's not going to be doing that. So I imagine that actually it's even slower when it comes to losing the capsule polysaccharide. So the timing, the amount of time that it requires for bacteria to actually lose this capsule polysaccharide is beyond when the mice completely clears away bacteria from its blood, from its system. So I think that's why there is no like larger, like more immune reaction in a short term. And then kind of like this is like part where we are not still sure, but like our kind of hypothesis was that, well, if we can make this bacteria lose this capsule polysaccharide, once it colonizes the tumor, then once it starts leaking out, they're going to be cleared more easily, so it will be safer. And it was quite surprising that we actually did see that. And then the fact that we can actually increase the dose by 10 times was very surprising even to me. And I'm sure there are other factors, but at least right now, that's kind of like our intention was to design this type of system where by controlling this capsule polysaccharide, can we improve safety? And then we see that it does. But I think that, again, kind of going back to my comment about this efficacy, like why more bacteria actually more effective, we're not completely sure. And then I think that even in the delivery and toxicity side too, I think there is a lot that we don't know. And then that's kind of like, from scientific perspective, I think that's very exciting that we can learn a lot more from this system.
I see.
So actually, as soon as you're talking about polysaccharide, I kind of thinking about my experience in company, because my job is basically making the high hyaluronic acid at that time for the biomedical application. But the most challenging part is not the production, because bacteria produce a lot, because we kind of engineer the bacteria, and then the real problem is removing the endotoxin below the whatever the level the FDA basically allow. So that is the entire the purification step, and that's very challenging. But we deliver, and then we finally got the FDA approval. But when as soon as you said, I mean, the polysaccharide, like immediately thinking about application for biocontainment. So I thought you're talking about, okay, make bacteria more vulnerable. In that way, if you kind of make the bacteria kind of lose all the polysaccharide, and then you can make the biocontainment easier. But you kind of using the different goal. I mean, so I kind of a little bit interested in that kind of. But have you ever thought about kind of using that system like for the biocontainment, so killing bacteria or making bacteria clear easier after all the cancer cells just die, something like that?
Yeah, I mean that is kind of the idea. So once the bacteria are in the tumor, they lose the capsule polysaccharide, so they become vulnerable. So that if they leak out, it's essentially biocontainment, right? Now again, by the survival.
But that's very the very delicate, sophisticated kind of tuning, because you need to kind of balance between the killing of the cancer, but if that happened too early before they kill the, so what I'm talking about is control the polysaccharide synthesis or losing synthesis ability after whatever signal, I don't know what kind of signal that they have. I mean, the cancer die, and then there are some signal, and then that trigger losing the polysaccharide. That also my original thinking as soon as you're talking about polysaccharide, but you kind of going in a very, very interesting direction. So I a little bit uh, confused at the same time that sort of works. So I kind of always surprised as well. So
Yeah,
that's
I mean, yeah, I definitely hear you, and I think that would be interesting if we can actually develop like a, say, like whatever, like maybe capsule polysaccharide system that can actually allow the bacteria to like survive in like immunocompetent like organism, or even mice, because mice actually have a pretty forgiving environment for bacteria, I think, systemically, compared to human at least. But like even then, as I said, like bacteria gets cleared from the system in like order like 1 to 2 hours. Like what you're talking about might be interesting if we actually develop this like super bug. Super. I want to be careful with language, but like this bacteria that can actually like sustain in healthy immune environment outside of a tumor microenvironment, then like that's something that we kind of might want to think about. But yeah, right now, the immune system is like so effective that even with capsule polysaccharide, even though it can give kind of competitive advantage of like a little longer circulation, for example, but it's still quite short. So I think that maybe a kind of like a useful thing would be like thinking about, especially also in like cancer patients, a lot of them are immunocompromised. Maybe in that case, like we can think about, even with a weakened immune system, if this kind of system can make it so that bacteria are really, really prone to uh, immune clearance, or yeah. And I'm thinking like not just like polysaccharide, but like, I mean, we do a lot of things in the lab and also like in the field, right? Attenuation. That's something that I'm thinking about. I think that's kind of like what you're alluding to, but can we have a biosensor to control the attenuation of bacteria, right? Because earlier I was talking about this clinical trial that eventually failed because perhaps because this bacteria was like overly attenuated. But can we control the attenuation level based on the kind of environmental factor? Maybe inside the tumor, we want them to be actually like very immunogenic too, as well as like survive better. But once it doesn't sense the tumor signal, they become really weak, and then they also become very less immunogenic, so that they're not toxic, and then they also get cleared much easily. And I do think that there is I think that goes back to kind of like our thought about, can we make this safe enough that it will be more acceptable to the patient and society, and then also practitioners, right? And I think that there is a potential in that direction, I think.
Sure. So now I realize I promised to let Dr. Kim go. Now I see the 2:35 a.m. Korean time. So let's continue if you could stay, but let's close officially and then we chat, after we, stop recording. Also, I kind of really delighted. This is the first time, as I mentioned at the beginning, first time 3 participants are Korean, and then it is the first time any. Let me think. I think the first time 4 participants kind of in the recording, and then interesting 4 of us is also Korean. So I believe,
Korean like
one thing I want to say, I'm very proud of being Korean, even though I'm
Korean one.
living even though I'm living in United States more than 20. Korean is crazy, yes, that's true as well. More than 20 years in America, but I'm very proud of being Korean. So let's continue the conversation, but I officially close and then stop recording, and then we talk about more crazier, topic after that.
Okay.
How about
Thank you.
that? All right. So,
Sounds great.
yep. So thank you. Also, this is longest kind of, recording, I believe, probably 1.5 hours. This is probably the record of the entire almost 5 years of series. All right. Thank you all for joining and staying today. We'll meet again on January 29, Thursday, the same time, the same Zoom link, but I'm gonna be in United States, so more convenient time for me, not the 2:30 in the morning. We'll have Professor Dina Petranovic at Technical University of Denmark. What a coincidence, because the Professor Kim also spent a couple of years in that university, and also Dr. Michael Grom at Yale University. As usual, the follow-up informal chat will occur without recording, as I mentioned earlier. So we already have the one audience joining us. So I'll basically, we'll continue to chat. Thanks. I stop recording. Just 1 second.